in some of the worst bombed areas, there's no dignity of a funeral.
Okay, this is a video, short video. I'm going to name it Molecular Various Compound Bioresimulation Measurements. This is pretty much everything that biology does, and I'm going to say I've been figuring this for a long time and studying medicine for between 7 and 10 years. Plus, I study physics and engineering and stuff like that, so I understand, quantum physics a little bit. I'm pretty decent at it. It's nothing but mathematics if you really want to show what the potentials are. That makes it most complicated. But I'm going to say I've been figuring for a long time. My name's Leonard Dance Jr. And this is how you, according to most common science that we know right now, you extract whole DNA, chop it up with enzymes or acids, or there are other processes like supersonic motion and things like that, or even gamma rays or X-rays, or some kind of chopping process. You run it through electrophoresis mass time measurement. Transfer to glass, validate with electron microscope. Simulate by restriction, enzyme break per size of section of DNA, compared to various break sizes of total DNA mass measured by time of flight mass spectrometer. Or just break up DNA into various lengths with enzymes and or acids, run them with PCR, copy them, maybe tag them, run them through electrophoresis to see the gene difference, and separate, run them through the time of flight mass spectrometer sequence spectrum of masses to the order of the DNA helix shape. If there are DNA segments large enough to give spectral identity, and identity is important in everything, especially atomic and molecular and compounds, to the mass shape identity of the entire DNA strand. Each mass will tell you the order of nucleotides as long as repeats are eliminated or accounted for. Or compared by separation by electrophoresis, and size of DNA fragment mass is large enough to eliminate section figures of the total DNA, then the whole DNA can be measured with computer simulations of the mass bonds in a helix figure. This is exactly how you measure DNA. There's no other way to do this for the most part. Not even X-ray crystallography or gamma ray crystallography or proton or electron. This is how you do it. This is like the only way. There's not another way to do this for the most part. It is a hell of a simulation. And other cell parts may be sequenced similar to this, especially with computer simulation. Chemical medicine sequencing is much of simulating all the simulations against a chemical reaction algorithm with energies, different kinds of energies. And that is pretty much molecular various compounds bioresimulation measurements. That's what you do when you want to learn how to heal the human body and defeat death and prescribe a medicine. You have to compare all of the molecular structures, the sequences, all the compounds, and it's a lot of sequencing, but that's how you do it. You do it with spectrometry, basically. You may also use a bit of chromatography, electrophoresis, time of flight mass spectrometer. You can actually use a little bit of X-ray crystallography if you need to. There are other processes probably. That's most of it. There may be specific gravities that you may need to measure. There's a lot of things you can do. Basically, you're going to use any tool in your tool shed that you can find. But this is basically the most scientific way of doing it based on all surrounding values that we have as a tool in the molecular/compound measurements of a systematic cellular process or multicellular systematic process. So it can't really be any different. There's nothing to use as an energy based on how the energy migrates with identities and how you collect an identity. An identity is what we're looking for. Most physical processes are looking for the identity of something or some kind of function. So, I mean, it is a mathematical identity. That's the number we want to reach, which identifies something, whether it be an atom, a molecule, a compound, or an entire system. And if you do the thinking, this is exactly the only real true way, for the most part, of really figuring out how to do bioresimulation. So this is my subject on laboratory and biolaboratory. This is pretty much it. And thanks for watching.